Lab diagnosis for sickle cell disease, because sickle cell disease is a component of chronic hemolytic anemia, and morphologically the cells will be normocytic, normochromic, with the long curved sickle cells. And peripheral blood smear will show polycytosis and isocytosis, sickle cell, target cell, and nucleated RBC, sometimes with inclusions like whole gel bodies and Pappenheimer bodies. And of course, because of compensation, there will be polychromasia, which means high reticulocyte counts, and also increased RDW because of compensation and the presence of multiple population of RBC in peripheral blood. Apart from that, there would be also other findings which affect leukocytes. Neutrophilia will be present, thrombocytosis, and elevated bilirubin. So bone marrow examination will show erythroid hyperplasia because of compensation, of course, and compensation of anemia will bring reticulocyte and nucleated RBC in general blood circulation. So in blood circulation, there will be also the presence of nucleated RBC, and also reticulocyte count will be high. So that's how it should look like. And the hallmark of diagnosis of sickle cell disease will be the presence of target cell and sickle cell. Okay, the hallmark of diagnosis of sickle cell disease is the presence of target cell and sickle cell. And you can see a number of erythrocytes and even spherocytes. And target cells, some of them are looking like echinocytes, but not. These are fragmented RBC. And also, this is a nucleated red blood cell, and a number of banana-shaped cells, like sickle cells. So the presence of sickle cell and target cell is a hallmark in diagnosis of sickle cell disease. So lab diagnosis can be performed in 2 steps: detecting insoluble deoxygenated cell, confirming the presence of them by performing electrophoresis. Someone is asking a question: which are the target cell in that smear? Oh, sorry. Let me show you the target cell. Let me show the target cell in the smear. This one, and this one. This one. You see? Target cell, they have like a target. They have accumulated hemoglobin in the center. And also at the periphery, it seems like the central pair has moved, and the center of the cell now is accumulation of hemoglobin. Like here, even like here, this one is target cell, huh? Very typical target cell, and others also. So the presence of target cell, these particular cells, and also the banana-shaped cells here, that is what you call, that is the hallmark in diagnosis of sickle cell disease. Thank you, Esther, for the question. Right. So for us to make a complete diagnosis of sickle cell disease, we can perform both solvent testing, electrophoresis, and high performance lipid chromatography, and also capillary electrophoresis. Sometimes we can also do isoelectric focusing, mass spectrometry, and DNA analysis to identify abnormality in genetic abnormalities. The hemoglobin solvent test is the most common screening method, especially explorating the hemoglobin's decreased solubility when is deoxygenated. So this is a very common practice in many labs, what we call solubility testing. What we have to do is just to mix sodium hydrosulfate, which is known as thymine, and saponin, which lies RBC and release hemoglobin. And saponin will dissolve the lipid membrane, and the thymine will reduce iron to the ferric state, forming deoxygenated hemoglobin. And deoxygenated hemoglobin polymerized and cause turbidity, while non-cycling hemoglobin solution will be clear. So this is how we do perform hemoglobin solubility testing. And as you can see, the left one is negative and the right one is positive, because the right one is insoluble. This is to detect cycling of RBCs, and that will be manifested in this solubility or insolubility of the tube. Because of deoxygenated cells, hemoglobin will be insoluble after being deoxygenated, and that can be detected as turbidity formation in the tube. Okay, secondly, we can also perform hemoglobin fractionation, which is also known as hemoglobin quantification. Hemoglobin can be quantified using electrophoresis or HPLC. And in electrophoresis, of course, it is to separate hemoglobin in accordance to their molecular charge. With negatively charged hemoglobin migrate towards anode in alkaline pH. All hemoglobin will be negatively charged when you use alkaline pH or cellulose acetate electrophoresis. But now this time we are using agarose or citrate agarose electrophoresis, which separates hemoglobin very well, because hemoglobins some of them will assume negative charge. They will migrate towards anode. Others will assume positive charge. They will migrate towards cathodes. And then we can be able to separate hemoglobins in their fractions. For example, hemoglobin S migrates together with the D and D, and C and E and O when you're using cellulose acetate. But when you use agarose gel, agarose electrophoresis, they will be separated very well. They will be separated, and you can be able to see hemoglobin S separately from DG and C. So this is what we will have to find. So it may not look like this, but this is a typical finding of hemoglobin. You can see we have cellulose acetate, electrophoresis, and also the citrate agar. Citrate agar is a acidic electrophoresis, and the top one, which is cellulose acetate, is alkaline or pH of 8.4, which is alkaline electrophoresis. And the migration of this molecule is like this, from here all the way, from left to right, from left here to the right. Because as you remember in the previous slide, we have said that in cellulose acetate alkaline electrophoresis, hemoglobin will assume negative charge, all of them, and they will migrate toward anode. And anode is this charge, positive charge. And they will migrate from here all the way to the right side. So which means that hemoglobin A migrates faster, the fastest, followed by F, followed by SDG, followed by A2, C, E, and O. So which is known as OR. So now, but when you want to separate, because as you can see, hemoglobin S has migrated together with the D and G, you may not know what this band represent. Whether this band represent S, whether represent G, whether represent the G or D is impossible. But you can only know that when you run citrate agar. So that's why you can see hemoglobin S migrates alone now. It is separated from others. And also C is migrating alone. And initially it was migrating with A2 and E and O. Let's see how we can interpret in your exam. Sometimes in your exam, you are given such question like this. And what can surprise you is that they can just remove these things and they give you this picture. And maybe they'll put patients here. Patient A, patient B, patient C, patient C, and so on. And maybe they will ask you patient C is representing which condition of the following. Okay, for example, let's try to interpret. This one is the, okay, let's check here at the cellulose acetate. For cellulose acetate, as you can see, this patient is having sickle cell trait. Sickle cell trait. Sickle cell trait is when you have heterozygous, one parent with the sickle cell, the second parent normal. So they will give you what you call sickle cell trait. It means you will have a normal percentage of normal hemoglobin and another percentage of sickle cell hemoglobin. So it is 40 to 60. 40 abnormal, 60 normal. So then it means you will have hemoglobin A, which is normal, and hemoglobin S, which is abnormal, because you have sickle cell trait. So from here, you can see this band represent A, and this band represent S. Okay, you will have also a small band which represent A2, for example, because here we have A2, a small band, because you know A2 is very small amount, like 3%. So that's how you can know this person is sickle cell trait. The person AB is sickle cell trait because you have hemoglobin A and hemoglobin S as they are migrating from the initial application point, which is here. You know how we apply sample. This is sample 1, sample 2, sample 3, sample 4, sample 5, sample 6, sample 7, and so on. So, and you can see we have different types of hemoglobin as they are migrating. So, but if we want to separate, to distinguish between the SDG, because as you can see, hemoglobin S migrates together with the D and G, and this is true of them, because when you want to separate them, you have to use a citrate agar. So, for example, let's consider our hemoglobin sickle cell trait disease. Sickle cell trait. Sickle cell trait, we have talked about hemoglobin S and A. First of all, we understand that in sickle cell trait, we have hemoglobin A, we have also hemoglobin A, and we have hemoglobin S. So when you want to separate hemoglobin S from these other hemoglobin, you have to run citrate agar, and you can see that hemoglobin S has migrated alone. It is alone, migrating alone, and you can see from the point of application, it is migrating towards anode, but hemoglobin A is migrating toward cathode. So which means that now on citrate agar, hemoglobin A and F, they assumed positive charge, and hemoglobin S assumed negative charge, plus C. C also is negative charge, and they're migrating towards anode electrodes. And you can be able to know this person is actually sickle cell trait because of the band of S and the band of A. So maybe we'll discuss questions in class when we finish this section of hematology, erythrocyte disorders. I will bring some questions where these things apply, and you'll be able to understand